All right, let's continue and talk about active transport. So active transports are they're all for carrier proteins, right? Because facilitated diffusion is when you can have channel proteins, and channel proteins, they just open up. So molecules will naturally go from high to low concentration, which is not what you want for active transport, because you have to go against the concentration gradient, low to high concentration in this case. And if you want to go from low to high concentration, this is going against the law of entropy, right? You're gaining orderness instead of becoming more and more disorder, right? So you have a cell, for example, you want more and more glucose in being pumped into the cell. When you already have glucose in the cell, then you have to go through active transport. So this process also requires ATP. All right, let's watch this video real quick. All right, so you can see this right here is our transport protein. It is right now closed, or it's inactive. So I don't know if you can hear this, the sound of this video, but it's talking about this word pump. So when we are, all right, so it's low to high concentration, right? This molecule will bind to the carrier protein, inducing a conformational change, right? So you saw that change in shape of the protein, and then it's able to release this molecule to the other side of the cell membrane. On to the next. Whoops. On to the next, right? So this right here shows you ATP. We're going to get more into it in the next unit. But when we think about ATP, we have this 3 phosphate groups, right? And when this last phosphate group breaks off, it's going to release energy. So when ATP is working alongside with a carrier protein that's in charge of active transport, is able to use that energy to cause this transformational change and help the molecule to get from the side of the cell membrane with low concentration of the molecule to high concentration of the molecule. This word pump is really important. When you use this word pump a molecule from one side to another, you're assuming that we're going through active transport and it's low to high concentration or against the concentration gradient. So this right here shows you a very typical classic example that we're going to meet again in the future. Is called a sodium-potassium pump. So this pump, this active transport, is very fancy in the sense that it doesn't just transport one type of molecule, it transports 2. So in this case, it will transport sodium ions from the cytoplasm to the extracellular fluid. So it's pumping the sodium out of the cell. And then it will then also pump potassium ions into the cell. So then what it's doing is that it will set up this concentration gradient of high potassium ions inside the cell and low potassium ion outside of the cell, and the opposite of sodium. So it's active transport in both ways. So we could watch this video real quick so you can see how this works. All right, we have the sodium-potassium pump. So this is active transport. We have sodium, this is the inside of the cell, coming in, 3 sodium binding to the pump. ATP breaks off, giving that energy to allow this transformational change. And then sodium ions, 3 sodium ions are released outside of the cell, even though there's already higher concentration of sodium outside of the cell. And now we have 2 potassium ion coming into the protein, all right? And then when this phosphate falls off, that induces another conformational change that allows this carrier protein to open to the inside of the cell. And then the potassium ions will be able to get pumped to inside of the cell, even though there's already high concentration of potassium inside of the cell. So now you have higher and higher concentration of sodium outside and higher potassium on the inside. This is a sodium-potassium pump. Real fancy. Okay, moving on. So we're going to skip this video. Moving on. So membrane potential is something that we're going to talk about again in the future as well, but it's talking about the difference in charges across the membrane. So what we mean by that is just like when we say hypotonic, hypertonic, isotonic, it's a comparison. So this one is a comparison of voltage on one side of the membrane to the other side of the membrane, right? So you know when you measure I think in the physics, you can have two clips, you clip onto two things, and then it will measure the difference in charges. That's what this is talking about. Right? So for example, negative charge in the cytoplasm would then favor movement of cations, so those are positive charged ions, into the cell if given the chance, right? So membrane potential is set up in this case when we talk about the sodium potassium pump. It will allow a higher positive charge outside the cell because you pump 3 positive out and 2 positive in. So even though both of the ions, both the potassium and the sodium ions are positively charged, because you're pumping 3 positive out and 2 positive in, as a result, you will have a higher positive charge on the outside and then a net negative charge on the inside of the cell. So that's called a membrane potential. Here's another word called electrochemical gradient. So when we talk about down the concentration gradient, right? So let's talk about the chemical gradient where you're just looking at the concentration of a certain molecule. But when we talk about electrochemical gradient, that's when ions are involved. So you're looking at the ion concentration and the membrane potential, so the charge at the same time, right? So for example, again, if you look at, let's go back and look at this picture right here, right? So we're pumping more and more of the sodium outside. So there is a chemical gradient of sodium ions, a higher concentration of the sodium ion outside the cell and lower concentration of the sodium ions inside of the cell, right? So that's a concentration gradient. But then there's also the electral gradient, or concentration gradient was a chemical gradient, right? Concentration of the chemicals. And then there's also an electrical gradient being set up because you're pumping more positive ions outside than there is to the inside. So there's a net positive charge on the outside and a negative charge on the inside. So that's a chemical gradient being set up as well. So you're no longer setting up a difference in concentration of the different types of molecules. You're also setting up a difference in charges across the membrane. Okay, so that's when we talk about electrochemical gradient. All right, moving on. This is a sodium potassium pump as well. We're not going to go way into it. We'll talk about it again in the future. Okay, this next one is called a proton pump. So a proton pump is specifically talking about hydrogen ions. So we can call hydrogen ions protons because hydrogen ions only has one proton. Okay, so that's a proton pump. The word pump tells you that this is, again, an active transport, low to high concentration gradient, low to high concentration of the molecule movement, so you'll build up more and more hydrogen at a certain part. So in this picture, you can see more and more of the hydrogen ions being built up outside of the cell. Okay, so this one is, again, an electrogenic pump because it's not only setting up a concentration gradient, it's also setting up an electrical gradient, because if you're pumping a lot of the hydrogen ions out of the cell, you will set up a positive charge on the outside of the cell compared to the inside of the cell, right? So in turn, you will have a net negative charge on the inside of the cell. Okay, so if you look at this little GIF over here, you can see the hydrogen is being pumped outside. Lastly, we have something called, second to last, we'll have something called cotransport. Let me drink some water. Cotransport is when two molecules are being transported together. Okay, so this is how this works. In this picture, you can see the proton pump. So hopefully by this point, you understand what I meant by proton pump. So it's pumping the hydrogen against this concentration gradient. So low to high concentration, you're building up more and more of the hydrogen ions outside of the cell, as you can see, right? So this is an active transport, so you need to use ATP as well, as you can see. Right, low to high concentration. So now you have this chemical gradient, but you also have this electrical gradient. So then given the chance, right, if you open up another door to allow these hydrogen ions to go back into the cell, it will do that, right? First, it's going to follow the concentration gradient. It will follow high to low if given the chance. If there's a channel being opened up, the hydrogen will do that, right? But there's also the positive and negative charge. The hydrogen ions will naturally want to go to the negative charged side of the cell because it's positive, it's attracted to the negative, right? So hydrogen will do that. just given the chance, right? But the really good thing is that when hydrogen is flowing back into the cell, hydro low concentration